Thanks, Jan. That was a great overview. Now, the use of biomarkers to enrich clinical trials is certainly a great idea, and there's at least 2 or 3 FDA guidance documents that kind of touch on the subject. But that doesn't necessarily mean that if you use a biomarker to enrich a clinical trial, you're going to end up with a companion diagnostic, right? Because not every single trial that gets enriched through the use of biomarkers ends up with mention of the biomarker in the labeling claim. So in the field of oncology, you talked about 20 drugs that have that companion diagnostic and that are approved. Do you have a sense for how many other drugs that don't necessarily have that companion diagnostic that were approved, but yet used biomarkers to enrich those clinical studies?
In fact, FDA has come up with a new type of definition. They called it complementary diagnostic. We heard it also yesterday from Pasteur. And they have used that in relation to the immune checkpoint inhibitors, both PD-1 and PD-L1. And of course, they use these biomarkers in relation to their drug development. But in fact, there's many reasons, I think, why they wasn't included in the drug labeling. If you, for nivolumab, for example, I would say that they didn't do their homework very well in relation to estimating the assay cutoff. And in fact, we have a new publication underway, in fact, discussing exactly this aspect. And so it do not seems like. There was one study that showed a clear trend that the one that had high expression of PD-L1 within non-small cell lung cancer that has an increased efficacy. The other one there, they couldn't demonstrate the predictive potential. So it of course has to do with the biomarker hypothesis. How strong is this? And I think that's also go into the evaluation by FDA when they say this should be a companion diagnostic and this should be a complementary diagnostic. But I think also, to be honest, I don't think that BMS was initially too interested to have a companion diagnostic that would restrict the patient population that would need to be treated with this drug. And this turned out to be a major downside for them because they have lost the first line treatment to Merck. So I always say, let science go first, not business. And then you can do good business. And I think that people at BMS, they will regret this. Sorry. Yes?
Thank you. This is a great presentation. This is rather a comment rather than question. So there are a few major differences between oncology and antibiotic in general, not just TB, because most TB drugs are repurposed drugs. So one of them is the fact that in field of oncology, you really talk about true companion diagnostic, which guide therapy from the get-go. In the case of other antibiotics, that really is not the case. We're talking about in vitro diagnostics. It becomes available when it becomes available. So at launch, you don't have to have a diagnostic. It will just treat empirically. The other difference is the time of the method of, say, the path of development. Here you have a parallel development between drug development and diagnostic development. In the case of antibiotic, that's not really the case. You need to get your approval first, get a breakpoint before the device manufacturers can actually attempt to come up with a device. And usually there's a big gap between those two, between the drug approval and then the device clearance. I think the agencies are doing a good job now in narrowing down that gap, which usually is due to review time and also the release of the data from the drug sponsor to the device manufacturer. So those are other differences I wanted to point out.
But as a comment to this, I would say we have also a couple of examples from oncology where you, after finalize the drug development, can come up with a companion diagnostic. KRAS in relation to a couple of antibodies there for treatment of colorectal cancer is one example. So, and also in relation to EGFR, initially it was immune histochemistry assay that was developed. It turned out not to be predictive, but you should look for mutation instead, and then the PCR technologies came, and that was also after approval. So I think that's a possibility also within other disease areas, that a companion diagnostics can come after the approval of the drug. And the regulators, they accept it. It might be more difficult with the company because you can see it's restrict, also to some extent the uses of the drug to use a companion diagnostic.
Do you think, a follow-up question then, do you think there's I want to say prefer, there's an advantage to the oncology for getting their devices reviewed faster?
Yeah, I think if you are within a high unmet need medical area, I think it's no matter if you are within oncology or if you are with another disease area. I think you will have these advantages, and you come with some really innovative. So I think that's something that the regulators are open to discuss. There might be people that has more experience on that. I know there are people from FDA, and they could comment on that. But that's how I at least see how the regulators look at these things.
Hi, I'm Dan Everett with the TB Alliance. I just have a practical question. In these, say, 20 cases or whatever, when the diagnostic is developed, is the actual platform method, the diagnostic test, is it in high-income countries generally available out in multiple hospitals where the patients are taken care of, or do I send a piece of tissue to Abbott Diagnostics in Illinois or wherever they are, and they do it centrally?
They use both strategies. So I would say, without knowing the exact figure, but might be 2/3 of them is assay that is distributed general to local hospital, to the local pathology labs. But there are also this central strategy used where you used a laboratory developed assay that you only can use in this specific laboratory, where you have to send your tissue sample or blood sample or what it is to this central lab. Yes.
No, that's helpful. And I suppose one difference is it's a little cheaper to send a tissue sample than
Yeah.
an infectious TB sample.
Yeah. Yeah, thank you.